So today we're going to talk about DNA replication, but before we get there, we're going to do a quick little review from yesterday about DNA and RNA, okay? So yesterday we said DNA has to be able to do the two following things: store hereditary information and be easily and accurately copied. So today we're going to talk about that second piece, okay? So we also talked about what DNA actually is, okay? The structure of DNA. So DNA stands for deoxyribose nucleic acid, and deoxyribose is the what that we find in DNA? The sugar, right, okay? So DNA is super important because it is what contains the directions for making all of the proteins in our body, okay? And proteins are what control everything, okay? All right, so DNA, we said, looks like a twisted ladder. So if we were to take a ladder that was flexible and we twisted it, okay, that would give us our twisted ladder shape. But DNA is actually described as a double helix. And that's, I like that you brought that up, because normally in a non-COVID setting, I would be doing a lab today with Twizzlers and marshmallows and toothpicks. Okay? Did they? So I usually do that lab, and I usually also do a strawberry DNA extraction lab. It's actually really cool.
We can't eat this class with food. I'm just canceling.
So, well, we'll see. I'll see what I can come up with this weekend, because if we all do our own, it might be able to be a thing. Okay, I'll see. All right. So the last piece of DNA that we talked about yesterday was what DNA is made up of, and the building block of DNA is a nucleotide. Okay, and a nucleotide is made up of three things. But when we're talking about DNA specifically, it is made up of deoxyribose, a phosphate group, and the following nitrogenous bases, A, T, C, and G. Okay? We also talked about base pairing rules yesterday. What always pairs to what? A to T and C to G. And how did I tell you to remember it? Beautiful. Okay, apples in the tree, cars in the garage. Okay, so those are our base pairing rules. We also talked about with the structure of DNA that the backbone, the sides of the ladder, the sides of DNA are made up of alternating sugars and phosphates. And in the middle, the rungs, those are made up of the nitrogenous bases or nitrogen bases and held together by a hydrogen bond, which is very weak, and that will make more sense today. The reason for replication, okay? They have to be able to break easily so DNA can replicate, okay? So those are our base pair rules, and we also learned yesterday that the order of the bases is what tells the cell what protein to make. And this is all gonna come together once we go through protein synthesis. Okay? Any questions? So that was all yesterday. All right, so today we're going to talk about DNA replication. What's another word for replication? To make a what? Copy. That's it, to make a copy. Okay, so when a cell divides, how does it make sure that a complete set of DNA instruction is passed on to each cell? The answer is it happens during that, but it happens there, but the whole process is DNA replication, okay? Which happens in the S phase of interphase in the cell cycle, okay? So before a cell divides, it duplicates its DNA in a copying process known as replication, okay? So this process is what ensures that each cell has a complete set of DNA instructions. So, like we just said, this happens in the S phase of interphase during the cell cycle, okay? So up here, you have DNA, and then DNA will be split apart, and we're gonna go through the process of how this happens. Once DNA splits, we can then make a copy of each strand. So one DNA molecule, when it's copied, now becomes 2. So during replication, the DNA molecules separate into 2 strands and they produce 2 new complementary strands using the base pairing rules that we talked about yesterday. A to T, C to G. So if we were to make this complementary strand, like we did yesterday, some practice, A would pair with G, C, T, G, A, C, C, perfect. Okay, that's it. So there's a series of steps that we have to go through in order for replication to happen. Okay, and this process is carried out by a series of enzymes. We've talked about enzymes, and what do enzymes do? So they do what?
They speed up.
Yep, exactly. They speed up chemical reactions by doing what? Yes, nicely done, okay? So they speed up chemical reactions by lowering the activation energy, okay? So we're gonna talk about a few enzymes today. So the first of these, okay, is known as DNA helicase, okay? Helicase is the unzipper. So it unzips DNA by breaking the hydrogen bonds. So up here you see DNA. If we were to unwind it and make it look like that ladder again, okay, helicase comes in and it unzips that DNA. So it is what is essentially breaking those bonds in order for that molecule to be separated, okay? So our twisted ladder is now coming apart. The second enzyme is DNA polymerase. This is super important because it has 2 jobs, okay? The first one is that it adds complementary nucleotides to the old template. So if we were to break apart that DNA, okay, we were to break this apart, we're gonna add to this strand and to this strand. So helicase is what broke this apart right here. And now we have polymerase, specifically DNA polymerase, because we are going to see an RNA polymerase. Okay, DNA polymerase adds your complementary strands. Okay, so if we had, for example, AGCT, and we had to make a complementary strand, what am I going to put here? TCGA. Perfect. Okay, so polymerase is what is adding that complement. The last step is also DNA polymerase. So the DNA polymerase, once it adds those nucleotides and it completes that complementary strand, it comes back through and proofreads. What does it mean to proofread? English. To go over it, to check for mistakes, to check for errors. Okay, so DNA polymerase comes back through and it checks for errors. It checks for mistakes. It checks for typos. Okay, So a lot of times when DNA is being replicated, there is an error. So the polymerase comes back through to check for those errors so that they can be fixed. Okay? So helicase first, unzips, okay, breaks the hydrogen bonds. Polymerase comes in, adds that complementary strand. Polymerase comes in, and now it checks for typos. Okay? So this is just the process. So you've got your DNA strand. What comes in here? What enzyme? Which one first? Helicase. You got it. Helicase comes in, breaks those strands apart. Then the polymerase comes back through, adds the complementary strand, and then goes back through to check for errors. Okay? 3 easy steps. Unzip, add, check for errors. Proofread. Unzip, add, proofread. Got it? All right. So at the end, each DNA molecule that results from replication has 1 original strand and 1 new strand, okay? This is called the semi-conservative model. So what I mean by that, and please tell me if you do not understand. So this is your original DNA molecule. Everybody with me? And then we come back in and we add new strands, right? The complementary strand. So these blue ones are the original ones. The pink ones are the new ones. So we start with 1 molecule of DNA and we end with what? 2. We start with 2 strands and we end with 4. Okay? We're doubling. So when we're making a copy, we're doubling. So 1 molecule becomes 2. 2 strands become 4. 2 old, 2 new. Does everybody understand that? Does that make sense? Because we're replicating that 1 original molecule, so we still have 2 old strands from the original molecule, but we're making 2 new ones. Okay? So we're just going to talk this out loud. Normally, thank you, COVID, you would have whiteboards, and we do like a boards up thing. Okay? So you're going to tell me how we replicate this. So ATG would be what? TAC. Thank you. ATA. GCT. TCA. GTC. GDG. ACT. Tag. Hear it. CAT. GGC. GTC. All right. Tell me DNA or RNA. Single-stranded. RNA. ATCG. No, I want you to tell me DNA or RNA. DNA. This guy right here tells you that that's DNA, that T. Ribose. RNA. Double helix. DNA. Gupta. How do you know? That U. Remember, RNA loves